The goal of this procedure is to determine the mitochondrial membrane potential and levels of reactive oxygen species in primary cortical neurons using the fluorescent probes TMRM and H2-DCF-DA respectively. This is accomplished by first preparing stock and working concentrations of the TMRM and H2-DCF-DA. The second step is to incubate the cortical neurons with TMRM and H2-DCF-DA at room temperature for 45 minutes in Thai Rhodes buffer. The third step is to image the fluorescence intensity of TMRM and DCF in live cortical neurons using 515, 570 nanometer and 488, 515 nanometer excitation emission respectively. The changes in TMRM fluorescence will be monitored in the presence of the mitochondrial uncoupler FCCP, while the changes in DCF fluorescence will be monitored in the presence of hydrogen peroxide. The final step of this procedure is the collection, normalization, and analysis of data. Ultimately, results are obtained by plotting a graph that shows the relative levels of TMRM and DCF fluorescence intensity before and after FCCP and hydrogen peroxide treatment, respectively.
Hello, I'm Joanna Bakowska. Welcome to my lab at Loyola University, Chicago. Today we will show you how to measure mitochondrial membrane potential and reactive oxygen.